Hello, I'm Auni Nazifa binti Atan with matrix number 109110.
Hello, I'm Ibila Sharmin with my matrix number 110214.
Hello, my name is Corin Chang Pei and my matrix number is 110522.
Hello, I'm Monica Lilanyao with matrix number 110994.
First, 4 ml of distilled water was placed in 7 test tubes by using micropipettes. Next, 1 ml of Bradford reagent was placed into each tubes. And then, 50 microliter, 100 microliter, and 200 microliter of BSA protein standard were added in and were prepared in duplicate. One test tube is left as blank. Next, mixture in each test tubes were mixed using vortex mixer. All of the mixtures were then placed into a cuvette, including the one that was set as blank. The samples were then read from this concentrated and the absorbance for each was recorded. Sample with the protein as blank was used to zero the machine. For part 2, the same procedure was repeated by replacing protein solution to unknown solution A, B, and C. 100 micrometer of unknown solutions were placed into each tubes in duplicate. One test tube was left as blank. Next, mixture in each test tubes were mixed by using vortex mixer. All of the mixtures were then placed into a cuvette, including the one that was set as blank. The samples were read by using spectrometer and the absorbance for each was recorded to determine the concentration of the unknown protein A, B, and C.
For the results in part 1, this table shows the absorbance values obtained when different volumes of BSA protein standard were added to the Bradford reagent and were measured using a spectrophotometer at 595 nanometer. After we obtain the data measured from the spectrophotometer, the results are then recorded in the table. Test tube 1 that does not contain any BSA protein standard is used as blank to zero the spectrophotometer. The average of absorbance and the standard deviation of absorbance for the test tube 1 is both 0. For test tube 2 and 3 that contain 50 microliter of BSA protein standard has an average of absorbance of 0.036 and a standard deviation of 0.02687. For the test tube 4 and 5 that contain 100 microliter of BSA protein standard has an average of absorbance of 0.0575 and a standard deviation of 0.01485. For the test tube 6 and 7 that contain 200 microliter of BSA protein standard has a higher average of absorbance of 0.099 and a standard deviation of 0.03394. From this graph, the x-axis is labeled as the volume of BSA protein standard, and the y-axis is labeled as the average of absorbance. We can see that the plotted points on the graph show a clear upward trend, whereas the volume of BSA protein standard increases from 0 to 200 microliter, and the average of absorbance also increases from 0.02687 to 0.03394. This shows a positive correlation, which means higher protein concentration leads to stronger absorbance readings. This is because Kumase brilliant blue dye binds to the proteins and changes color at the measurement of 595 nanometer. Each of the points plotted on the graph represents the average of the two readings for each BSA volume. The arrow bars at 50, 100, and 200 microliter represents the standard deviation of the readings. The large error bar represents the greater standard deviation and correlates to a more variation in the duplicate readings. We can see that the 100 microliter sample has the smallest error bar, which means a lower variation compared to the others. The 50 and 200 microliter have more variation as both have large error bars. Now moving on for part 2. To determine the concentration of unknown protein solution of A, B, and C, we use the formula derived from the BSS standard curve, which is y equal to 0.02x. By using this formula, we obtain the data for the concentration of unknown protein and recorded it in the table. Solution A has a concentration of 2.925 micrograms per milliliter. Solution B is 3.125 micrograms per milliliter, and solution C is 5.150 micrograms per milliliter. This is the graph of protein concentration against average of adsorbents at 595 nanometer. We can see that there is a linear relationship shown by the equation y equal to 0.02x and r to the power of 2 equal to 1, which indicates that the data is highly accurate. The linear trend in the graph shows an increasing trend that as the adsorbents at 595 nanometer reading increases, the concentration of the protein also increases.